Meiosis is the cellular process to get ready for making babies. It happens in men and women. It happens in plants and animals. It happens in produce and fungi too, but their life cycles are a little weirder. So we'll just stick to pretty much plants and animals, mostly with a human focus, and figure out how this works. It looks a lot like mitosis that we studied a while ago in terms of the process of pulling apart chromosomes. We'll mainly focus on the chromosomes themselves and how those get split apart in the end, rather than the step-by-step process. One of the key things about meiosis to be looking for is that it results in genetic variation. It's the reason that no two people on Earth look alike. And even siblings, even though they share the same parents, have basically no shot at looking exactly like one another. Okay, except for identical twins, but that's a different story. I'm going to tackle meiosis a little bit differently than usual. So these are the topics we'll go over. We'll mostly look at the animation on the screen. And I want to start with a review of life cycles to put this into context, and also chromosomes, because it's been a little while.
All right, remember that meiosis makes gametes, and those are only made in specialized reproductive organs. The female reproductive system here in purple, the ovaries, and in the males, the testes. In flowers, we only get it in the anthers and the ovaries as well. Everywhere else in the organism, it's mitosis that is the type of cell division that happens. So meiosis is far more specialized. In the human life cycle, meiosis sets us up for fertilization. Notice how the life cycle here is divided in half. We spend most of our time down here in the diploid phase, where we have two sets of chromosomes. Notice how there's two copies of chromosome 1, two copies of 2, and that goes all the way through all 23 pairs of chromosomes. But we do spend a very brief period of time as a haploid, and so let me use the more complicated version of the life cycle over here to sort of put it in this simplified format. So first of all, an individual over here who's diploid goes through meiosis, and that would produce, for females, an ovum. Obviously, for fertilization to take place, we need two individuals. The male goes through meiosis and produces sperm. So now those are the two haploid cells. Those cells undergo fertilization and produce a zygote. So that zygote is the first cell that is actually us, and it is diploid. It has two sets of chromosomes, and from there it just keeps going through mitosis and mitosis and mitosis, and also differentiation, development, until we get to adulthood and can repeat the cycle. So from a chromosome point of view, we have two sets of chromosomes. Here's one set, and you get another set. You get one set from mom and one set from dad, but you could only pass on one of those sets. But it's not just mom set or dad set. We have to shuffle them up a little bit.
Well, you remember that chromosomes come in pairs, and what we have here is a homologous pair of chromosomes. It's going to be important that you remember the difference between homologous chromosomes and sister chromatids. Homologous chromosomes, we got this chromosome from dad, and we got this chromosome from mom. They're similar, they contain the same genetic information, but different versions or different alleles. So that's homologous chromosomes. But sister chromatids, those are made, remember, during interphase, specifically during the synthesis phase. And that happens when DNA replicates. So there's a bunch of concepts from the past that we've looked at, all coming together here, and that produces these identical sister chromatids. And these down here are also sister chromatids. All right, so now we're ready to move on to just an overview of meiosis. You'll find it looks a lot like mitosis, but with some really important changes.
All right, here we start shoe meiosis. We start in interphase, really just before meiosis, and we need to, just like mitosis, make an extra copy. So those are the sister shoes or sister chromatids. Then we need to start moving them around. So we enter prophase 1. We need to get rid of the nuclear envelope, grow some microtubules, and this pushes together the homologous shoes or homologous chromosomes. Here we have them sitting right next to each other, right and left, same type of shoe. And that's where the analogy really comes in, is just like chromosomes come in homologous pairs of similar chromosomes. Here we have similar shoes, a little easier to see. From here on out, everything is kind of as you'd expect. We need to line them up. That would be metaphase 1, but notice they're lining up as pairs. Well, really pairs of pairs. We got all 4 of them together, these tetrad shoes. And then they get pulled apart in anaphase, as you'd expect. In telophase, we get the nuclear envelope back. Cytokinesis, we divide them. And now we're down to 3 types of shoes in each. Yes, we have 6 shoes, but there's really just 3 types. What good would it do you to have 2 right shoes? You can't put one on your left foot, so they are identical. So we have 3 in this cell and 3 in this one. Here we have a right, and a left, and the opposite in the other one. Moving on, we need to keep going, and in prophase 2, get rid of the nuclear envelope, start pushing things around, which takes us to metaphase 2. Now the sister shoes are lined up. They get separated in anaphase 2. Telophase, we get the nuclear envelope back, and cytokinesis, we divide them up. Now it's a little easier to see that we really have 3 shoes in every single cell. But what good would that do you, just having a right shoe, and a left shoe? Really nothing until you combine it with the left, and right of another individual's gametes. And there we really kind of see how this plays a part in the life cycle.
Well, let's look at that in a somewhat more formalized way. So here's all the major phases, and I'm going to note that there's a big dividing line. Over here we have meiosis 1 and then meiosis 2. And really everything interesting happens in meiosis 1, so that's going to be our main focus. But a brief overview, we don't focus quite as much on the phase names, but some critical ones are prophase 1 and metaphase 1, so be looking out for those in particular. So notice here in prophase 1, we're grouping up those chromosomes. Here we have 6 chromosomes, and they are coming together in homologous pairs. So you notice that you got a blue and a red together here, and here, and here. So that's where the difference between meiosis and mitosis really starts, is homologous pairs come together. And then homologous pairs line up. Then everything else starts looking familiar. They divide, and then they get pulled apart into 2 cells in that telophase. So at this point, once they divide and we enter the second part of meiosis, each cell only has 3 chromosomes, right? 1, 2, 3. So we've already reduced our chromosome number, but we still have those sister chromatids. So we need to divide those up. Otherwise, we're going to end up with double the amount of DNA that we need. So we line them up in metaphase, we pull them apart, put them in different cells, and our result is 4 cells, each with 3 chromosomes. And that's in contrast to the 1 cell we started with 6 chromosomes total. We've cut it in half. That's the main idea. All right, so with that brief overview, let's revisit the important parts. And all of them are in meiosis 1, starting with independent assortment. And that's when, in metaphase 1, those homologous chromosomes line up. Do they line up with the red on the left and the blue on the right, or they switch? And what happens to all the rest of those homologous pairs of chromosomes? Let's find out.
So independent assortment happens right here in metaphase 1. Independent assortment, remember that. That's what Mendel talked about. We'll come back to that in a minute. Looking at it this way, instead of using right and left shoes, we use blue and red. So when we line up chromosomes, like in this cell here, we could get blue on the left and red on the right. This cell down here is exactly the opposite, and the rest of them are really every possible combination in between. And I think it's worth kind of looking at the mathematics about this. You spent all those years in math, might as well pay off for something. Okay, so for this first pair of chromosomes, how many ways can we line them up? Obviously, just two. Either the blue goes on the left or it goes on the right. Same with this one. There's two ways we could line them up, and same with the third pair as well. So the math on that is 2 times 2 times 2, which is 8. Another way of expressing that we'll need to come back to is 2 to the third power. So all that is 8 possible combinations. Okay, so that's how you could figure out how many ways you could arrange the chromosomes during meiosis. Let's take that to the human example. So for humans, we have pairs of chromosomes, so there's 2 chromosomes, right? And we had how many pairs? Here we had 3 pairs, but we have 23 pairs. That's going to be a bigger number, probably even bigger than you expect. If you punch that into the calculator, you get somewhere around 8.4 million. That is a big number, just from lining up chromosomes differently. And here's what that would look like. Let's go back to our shoes. I like them better than the blue and the red. When we line them up this way, and we pull them apart, then you would get right, right, and left, or left, left, and right, depending on how you arrange them. But what if you switched them? Then you would get a different combination. Again, that's just for 3 pairs of shoes. For humans, we have 23 pairs of chromosomes, so we get a lot more combinations, which is one of the main reasons that you don't look anything like your siblings, because each of your parents have 8.4 million ways they can pass on their chromosomes. And this is where we finally get back to Mendel when he talked about independent assortment. He talked about this a half a century before anybody even knew about meiosis. And this deals with 2 traits. In this case, on the Punnett square, pod color and pod shape. And he really noted that there was different ways those could all combine, that you could get different combinations of those traits because each are carried on a different chromosome. That's what we know now. Do they line up with blue on the left and red on the right, or do they line up differently? And that is going to affect the outcome and how those traits are passed on and combine in offspring.